Welcome to another lecture on Virtual Microscope Simulator. In this video, we will be exploring the world of Virtual Transmission Electron Microscope Simulator. Transmission Electron Microscopy, or in short TEM or TEM, is a unique and revolutionary technique that enables us to visualize materials with unprecedented details. It plays a crucial role in advancing our understanding of materials, biological structures, various phenomena at atomic level. With its ability to provide high-resolution imaging and elemental analysis, TEM empowers scientists to unravel some of the most complex structures, investigate the chemical composition, and contribute to breakthroughs in the fields such as material science, nanotechnology, and biological research. However, the anatomy of modern microscopes are complex, and it is accessible to only a handful of researchers. Furthermore, the increasing complexity of the microscope is also a challenge for teachers, students, and researchers to understand the working principle. Digital tools such as virtual microscope simulators can overcome the traditional challenges by empowering learning, teaching, and training. We begin by visiting MyScope TM simulator website created by Microscopy Australia and Thermo Fisher Scientific. In the top of this website, you will also see the instruction how to proceed with various actions. In the right side, you will see an anatomy of transmission electron microscope and its various components. If you're interested to know what all these components role and what does it mean and how it functions, I have a separate lecture on explaining the components and its role and what they are made up of. In the left side, you will see the various actions that you have to perform and keys or knobs as you move forward with the experiment. You will start by removing the specimen holder first. Once you click the remove, so you will see the holder is now removed, and it says this sample icon is now active. So you can select a set of samples from the library that's already available. We can choose nanoparticle, and what you saw here is the sample TM grid is now loaded. And now the insert icon or specimen holder insertion is activated. You just click on that, and now it is evacuating here. Once it is evacuated, then you can insert the holder. The holder is inserted. Now you have to select a target voltage, that is accelerating voltage. In this microscope, it was designed for 200 kilo electron volt. And now once it reaches the target voltage, you will switch on the beam. And you see what changes occurs here, the electron beam, which is passing through the specimen, and then you have all the way the camera here. Now, when you do this, something else changes. What you see here in the background is already a TM grade, but the beam is off center. It's away from the center. So your role is to bring the beam to the center using this beam shift. Because it's activated now, so you can use it, and you can bring the beam and center the beam. And beam has to be centered in the yellow cross that is pointed in this place. You bring close to center using X, Y keys, and now it's already centered. And now it is activated here, the brightness. It means that you have to spread the beam all around this grid area. You are spreading the beam now. Now it says the aperture, so we can go and look into the diagram, condenser aperture. So you can insert the aperture. You can notice the changes soon. The insert icon is now activated. You can insert, so the aperture is inserted. Now, once you insert the aperture, you also have to choose the correct aperture size. It can be large, medium, small, so we can choose a medium. We can see some changes behind here, so we choose the medium aperture. Now something has changed here. What you see is the beam again moved out of the center. At the same time, it is also has some issues. The circular shape has changed into kind of an oval shape. Now first you have to center the beam. And this particular change is also called astigmatism. And you have to make, first center it and then make it astigmatic. So here is a stigmator control. So you can use X or Y. So when I use minus X, it becomes worst. If I choose Y, it only expands in one of the directions. So maybe we have to go plus X. And once it is circular, this yellow mark will disappear. And now again, we have removed the stigmatism and the beam is circular, but we have to spread it and increase the brightness. Now we are almost set and we have to choose a magnification where we want to see. The begin with a very low magnification. We already see here some particle behind, but however, we have to make sure that there is a minimum movement of the samples for which the wobbler is now activated. We can choose the wobbler and sample is wobbling quite much. So you can choose Z plus or minus to minimum, make it almost there is no wobbling. And once there is no movement, then you can just switch off the wobbler. Now it asks for us to look for a diffraction before we go, how the diffraction pattern look like. So you see here already a diffraction pattern. What we have to look now, because it is activated, the aperture icon is activated, we may have to insert something else that is objective aperture, and we can see the objective aperture is below the objective lens. So let us choose. Now it says that we can insert this. So let's insert this objective aperture and we can see what changes. So the beam has moved quite much away from the center. Now we have to bring it to the center using X or Y. So we are very close. It is now fine. Now everything is done. We can do a low magnification imaging. So here is the low magnification image. But what we have to look for, select an area where you are interested to take the image. Now it is highlighted certain area with a blue circle. It also has a blue cross. Before imaging, we want to bring it to the center using this stage position function or the icon. And the blue cross should overlap with the yellow cross. That shows the center of the viewing area. Yes, now it's matched. So we are in low magnification, but now we can go slightly higher magnification to medium. Once again, we have to do the same. We see a circled area where we are interested to look and take an image. This cross here should match with the cross sign, which is the viewing area. So we move it by X and then go a bit Y direction. I think we are almost there. Yes. Now, once if you are happy with this, you can take an image or you can continue for a higher magnification. So we are interested to have a closer look at this particular particle. So we move it to the center. One nanoparticle at the center so that we can have a better details. Yes. So now we are here. So what we have to do is that this particular particle looks a bit fuzzy or out of focus. Our goal is to make it more focused. So what we'll see is that objective lens, the objective lens is here, and this is the one which will help us to get a better focus. So we are very close. I think now we can see, in this particular case, we can see the lattice fringe. Now, before taking the image, we have to insert the camera. So let us go and see what happens when we insert the camera. The icon is active and the camera is inserted and you already see an image. So now we can go and acquire an image. Once you acquire an image, you can save this image. What next can be done after acquiring image? Once you acquire an image, it says that you can move to the next activity. The next activity here is the diffraction, or looking at the diffraction pattern of the particle. So we click on diffraction, and now it says the brightness is active. So we have to make it maybe smallest possible spot as, yeah. And now we have a very small spot, and we will have a look at the diffraction pattern, or we will switch to the diffraction mode. And before we do that, we can look at also the diagram here. And if there is, you see any change. And what we see here is a diffraction pattern. Now we have to move this to the center. So we take a specimen tilt, ember, okay. Yes. Now we can also have an imaging. So it's active. So yes. And now we have to spread this beam to the whole viewing area. Yes, now we have spread. Now it asks us for something. Because we are talking about diffraction, we have an aperture here, which is a selected area diffraction aperture. So we have to choose SAD aperture, and now this is active. So it's activated, we insert, and you can closely see what actually happens here. We have inserted in the background, we will see the beam has moved a bit out of center, so we can move the beam to the center. Once it's in the center, so this is active now. Now all that we are interested is look at the diffraction pattern of this particular nanoparticle, which is in focus. So if you see diffraction, this beautiful diffraction pattern is visible. Now, when I switch on the diffraction, and you also see that this beam stopper icon is activated, and you'll see also a blue color indicated here about the beam stopper. Because the beam is so powerful and it's strong enough to damage most of the digital camera. So by inserting it, we can prevent some of the central right area. So now we will insert this beam stopper. When we insert the beam stopper, we also see that the beam stopper is here. Now we can insert the camera if you're interested. So we can insert the camera. The camera is inserted. As you can see here, beam stopper inserted, and you have a beautiful diffraction pattern which you can collect. Now, once we collect the diffraction, it says whether you are interested to go back to the basic imaging or you want to move to the high resolution activity, HRTEM. So let us click on high resolution activity. And now we have to retract the camera first. Let us see what action takes place here. When we retract the camera is retracted, and we go to imaging mode. So we can go to imaging mode, and we still have the beam stopper here. So the beam stopper can be taken out. But before that, we have to take out the aperture, and then we remove the beam stopper. Because we are interested in high resolution, it means that we want to see individual atoms. Now this is relatively high magnification, but not high resolution. So let's go to very high magnification, where we are able to look at individual atoms. When you go high, again, the focus changes, and we have to play with the objective lens focus. And you can go either way, deep over or under focus. At some point, you start to see individual atoms appearing here. So almost atomic resolution. And at this stage, this is good enough. You can have a look at Fourier transform. So we can switch on FFT. And what you see here is it is not stigmatic, it's astigmatic. Now this is astigmatic, and we can proceed to the next step. We can switch off the FFT, and you will see again here the camera is almost inserted. Now we can go to high magnification, and we can go find the correct focus here, almost very close. And now we can acquire the image. When you acquire the image, again, it says whether you want to go to the basic imaging or you would like to continue to STEM mode, that is scanning transmission electron microscopy mode, where you will view some mineral sample as an example. So we could go and continue for STEM mode. And meanwhile, we can also see what changes occurs in the microscope. So let's go continue to STEM mode. And what we have to do here is first remove the condenser aperture. And now we have to switch to STEM mode. Many of the modern TEM microscopes, you don't have to have a long time gap between TEM and STEM. You can switch rather frequently. However, it's a good idea to switch off the beam when you change from TEM to STEM or STEM to TEM mode. Let us switch to TEM mode. What you see here is called a ronchogram. It is similar to FFT that we see in TEM. In STEM mode, it's a ronchogram. It also has to be circular. And now we can make it circular by using this objective lens focus. And there should be no pattern. So we have to achieve that target, that at the center or in the middle, there are not any pattern. Yes, we have reached very close. Now it says that insert a lens. It is a condenser lens. So let us look at here because we have removed beginning. So now it is activated. Now, when you switch on this, you will also see some changes behind. So here, and we have to move this to the center, ronchogram to the center. Yes, almost. Yes, now we see. Once it is done, it says that we can now insert the condenser aperture. Yes, it's inserted. Once you insert, you will see again it's off center. So we move the beam current bit to the center. Yes, yes. Now it is done. Once it is done, we have two types of STEM detectors. We can insert them. So here it's a bright field. So we have inserted. Now we can go for a bright field imaging. And let us click the raster button. So what you see here is already start to image. Now we can acquire the image. We can go higher magnification or very high magnification. What you see here is a really high magnification, an individual atom in STEM mode. Now, yes, we are approaching a good focus and we can achieve and see, acquire an image. Now you have done the entire sequence of event that can be studied using TEM. Low magnification, high resolution TEM, diffraction pattern, and scanning transmission electron microscopy. However, the modern TEM offers huge number of other possibilities, such as elemental composition, which is called EDS. We can also get elemental composition as a spectra, EDS spectra or EDS map. We can also study yield spectrum. We can study the optical property of the material in situ.